Hello guys, welcome to our channel AskPrep. Today's series is of 11th class biology. We will be studying about chapter 6, that is anatomy of flowering plants in detail. Also we had added important note points for your examination perspective. All the best for your exam. Let's start the video. Starting with introduction. Anatomy of flowering plants. Flowers are not just pretty things to look out other than their beauty and fragrance. They fulfill many more important functions. To start with, they are a reproductive organ of plants. They consist of many structures that help the plant to survive, grow and reproduce. Next topic is the tissue system. Plants are made up of cells which are grounded into tissues that perform specific functions. These tissues are further organized into organs with specialized roles. Each organ within a plant has its unique own internal structure. Tissues are categorized based on where they are located in the plant body. There are two types of main plant tissues: meristematic tissues and permanent tissues. Meristematic tissue. Apical meristem located at the tips of roots and shoots. This tissue produces primary tissues like dermal, vascular and ground tissues. Intercalary meristem found in grasses. It is situated between mature tissues. Lateral meristem responsible for generating secondary tissues like cambium. Permanent tissue. Simple tissue comprises a single cell type with a consistent structure and a function. Complex tissue made up of a multiple cell types that work together in a coordination. There are three types of tissue system based on their structure and location. These are epidermal tissue system, the ground or fundamental tissue system, the vascular or conducting tissue system. Epidermal tissue system. The epidermal tissue system is like that plant's outer coat covering its entire body. It's made up of different parts like epidermal cells, stomata and little half-like structure called trichomes. Picture it is the skin of the plant. This outer layer called the epidermis. It is made up of closely packed cells forming a protective barrier. Epidermal cells are usually thin and filled with a big bubble-like vacuole. They are covered by a waxy layer called cutelle, which helps them water inside, kind of like how our skin keep moisture in. Stomata are tiny holes in the leaf skin, allowing plants to breathe and release excess water. Each stomata has two guard cells that control its opening and closing. In grasses, these cells look like a bit different, more like a dumbbells. Surrounding the guard cell, there are some other special cells called subsidiary cells. Further moving on, ground tissue system. The ground tissue system is like the plant of a body, supporting and filling it out. It's made up of different type of cell tissues. Simple tissues like parenchyma, collenchyma and sclerenchyma. These tissues are the building blocks helping the plant grow and stay strong. You can find them between various parts of plants except for the skin and the plumbing. In the stem and roots, you will see parenchyma cell in the cortex, pericycle pith and medullary rays. These cells are like flexible, versatile workers adapting to different tasks. In leaves, the ground tissue is called mesophyll, where you find thin-walled structure full of chloroplasts, the plant green energy factories. Think of it as plant's flesh providing support, storage and energy production. Moving forward, vascular tissue system. The vascular system in plants is like their circulatory system, transporting nutrients, water and sugar throughout the plant. It made up of two complex tissues, the phylum and the xylem. Together these tissues form vascular bundles which are like the plant blood cells. In dichlorodinous stems, there is a layer of tissue called cambium between the phylum and xylem. These cambium can produce secondary tissues like xylem and phylum, making these bundles open because they can grow wider. But in monocyclodons, there is no cambium present, so they can't produce secondary tissues. These bundles are called closed. When you look inside the vascular bundle, you might see different arrangements. In radial bundle, the xylem and the phylum alternate along different radii like in roots, but in conjoined bundles, they are grouped together around the same radius. Anatomy of dichlorodons and monocyclodons plants. Dichlorodons and monocyclodons plants differ in their anatomical structure. Dichodes have two cyclodons, reticulate venation and a taproot system with the vascular bundles arranged in a ring. Monocots on the other hand have a one cyclodon, parallel venation and a fibrous root system with scattered vascular bundles. Let's explore the tissue organization, root, stem, leaves in more detail. Dicotyledonous root. Apilema, the outer layer of the root with the unicellular roots hair cortex, consists of a thin-walled parenchyma cells, endodermis, barrel-shaped cell with a Casparian strips lacking a intercellular space. Pericycle contains thick-walled parenchymatic cell in shades lateral roots and vascular cambium. Pith, small or incompacious conjunctive tissue, parenchymus cell between xylem and phloem. Steel, inner tissues including pericycle, vascular bundles and pith. Monocotyledonous root. Similar anatomy to dicot root with epidermis, cortex, endodermis, pericycle, vascular bundles and pith. More than 6 xylem bundles and compared to dicot root, large and well developed pith does not undergo secondary growth. Dichlocodoneous stem. Epidermis protective outlay with a trichome and stomata, cortex, hypodermis and middle parenchymatic cell and endodermics. Pericycle semi-lunar patches of sclerenchyma. Medullary ray radically placed parenchymatic cell between vascular bundles, vascular bundle. Arranged in a ring with a conjoint open bundle and an endark protoxylem. Pith, central portion with a parenchymatic cell providing structure, support and storage. Monocotyledonous stem. Scleromatex hypodermics provides mechanical strength beneath the epidermis. Numerous vascular bundles scattered throughout the stem, each surrounded by a scleromatic bundle sheath. Conjoint and closed vascular bundles, smaller peripheral bundles compared to centrally located ones. Absence of phloem parenchyma, water containing cavities within a vascular bundles. Doris ventral leaf, also known as dichlodoneous leaf, epidermis, covers both upper and lower surfaces with a small cutile more stomata on the abaxial surface. Mesophile chloroplast containing parenchyma cells between upper and lower epidermis, consists of a palisade parenchyma and spongy parenchyma. Vascular system, vascular bundle surrounded by thick walled bundle sheath cell visible in the veins and midrib. Vein thickness, varying thickness in a reticulate venation of dicot leaves. Isobilateral leaf. Similar anatomy to Doris ventral leaf, but the stomata on both epidermal surfaces. Undifferentiated mesophile, no differentiation into palisade and spongy parenchyma. Parallel venation. Veins run parallel in a monocot leaves. Buliform cells in grasses, adaxial pyramidal cells along with veins modify into large empty buliform cell responding to water availability by altering leaf surface exposure. Moving to last topic, secondary growth. Secondary growth, a process of characterized by the widening or thickening of a plant stem or roots, contrasts with a primary growth which occurs at the tips. This lateral expansion, typical in dicots and gymnosperms, lead to the formation of woody tissues. Key to secondary growth are two lateral meristems, the vascular cambium and cork cambium. The vascular cambium generates secondary xylem inward and secondary phloem outward, enhancing mechanical support and facilitating water and nutrient transport. Meanwhile, the cork cambium produces cork cells to replace epidermis, forming protective bark. The annual rings seen in tree cross-sections reveal growth patterns influenced by environmental conditions. Secondary growth is vital for creating robust and enduring plant structures. Thanks for watching video. We will be dropping notes in description of this video. If you found this video very helpful, kindly do like, subscribe and share. See you in next video. Again, all the best for your exam.